Hey, welcome to week 5. We're going to be starting a 2-week unit on population genetics. And this is the introductory lecture on what the terms are, the important terms are you're going to be hearing for this week and next week. So population genetics. First I want to start talking about this thing we call a gene pool. How can you think about a gene pool? Well, it's a collection of all the genes from a population. Usually we're talking about specific genes that we're tracking, and so you can think of it in terms of, like I like to think of like a swimming pool. So you're thinking about pools where you toss everybody's genes in there, and so the key thing is thinking about percentages. So we can think of how rare or how common a gene is in a gene pool, and we're watching those change over time, and that's the evolution. And I'm going to explain what an allele is here in a second. So there's two terms you might have heard before, genotype and phenotype. Let me just take a second to explain what these are. And you can see from the roots of the word, you can start to guess what they mean. Genotype is the genetic makeup of an organism. So we usually talk about genotypes like we talk about the genes that they have, what versions of the gene do they have. The phenotype then is how the genotype ends up being displayed. Sometimes we can see this. For instance, we can look at the color of somebody's hair. That's your phenotype. Now, it's a pretty complicated phenotype because it's affected by a bunch of different genes and even things that may not even really be part of evolution, like some of us actually go in and change the color of our hair purposefully, but that doesn't change the genes that give us our natural color. So genotype will be tracking the genetic makeup of organisms. Phenotype, how they look. So that base there of phenomenon is the look or how it's displayed. And if we're talking about genotypes and phenotypes, let's take a second to talk about two different terms you're going to hear a lot in population genetics, and that's genes and alleles. So down below I have an example of the image from the chromosome that carries the sickle cell anemia gene. So on chromosome number 11, there's a region of the chromosome that codes for the hemoglobin gene, and it's that stretched DNA that would tell you what the protein should be and what sequence the amino acids in the protein should be. So a gene in general terms is an area of, or a stretch of DNA that carries the code for a specific protein. In this instance, it's the protein for hemoglobin. And it's carried on chromosome number 11. So there's a bunch of genes on chromosomes. We're just looking at one specific one, hemoglobin in this case. So it codes for that specific protein. So every protein has its gene that we can go back and search for. So what's an allele then? An allele is the different versions of the gene. So for instance, below we have hemoglobin, but it comes in two different versions. It comes in the normal version, which they put a little super number or super letter A up by the hemoglobin gene. So they show it HB with the A raised up like that. And the other version for hemoglobin codes for a protein that causes sickle cell anemia disease. And that is shown down below as the gene HB again, and then they put an S in a superscript to indicate that version of the gene codes for the abnormal version of the protein. It's only one amino acid change in this whole string of amino acids that code that make hemoglobin that causes this specific disease. So genes, stretches of DNA, alleles, different versions of that DNA. Okay, so as you watch the video this week about humans and hemoglobin, you'll see some of these same terms used in that video. So another way we can look at this is most humans are called, or most organisms are what we call diploid. That means that you carry two sets of chromosomes. One came from the egg. Thank you, Mom. And the other came from Dad's sperm. So each of them contribute a full set of chromosomes, so you actually end up with two full sets of chromosomes. Then that di at the beginning of the word diploid, that means two. So most organisms are diploid. The alternative is to be haploid, which means that you only have one set of chromosomes. So we call eggs and sperm haploid because they only carry one set. And there's some rare cases of animals that are haploid, that they don't need to go through sex to actually produce offspring. They're strange. You can look them up if you want to. Haploid lizards. So when we look at diploid organisms, we look at their genotypes, there are going to be two versions of the alleles at sometimes. So homozygous refers to the two alleles being the same. So Mom and Dad both gave you the same version of the gene when they combined and created you. Heterozygous means that you have two alleles that are different from each other. So Mom gives you one version, Dad gives you another version. So if we look above at that hemoglobin example, you could have two normal hemoglobins, HbA, HbA. You could have, so the homozygous example will be something like this. Or it could be the other case, or you could be the other combination as well. You could be the HbHs. Oops, let me fix that for a second. Those are the two cases of homozygous for hemoglobin here. So you can guess now what the heterozygous combination would be. It's exactly what I typed up above if we were going to consider that's all in the same organism. One copy of the normal, one copy of the sickle cell version. So that brings us then to What does it mean to be dominant and recessive? You may have heard this before in discussions about genetics. So the dominant gene isn't the one that's in the biggest percentage. Many times the dominant gene is the largest percentage of the gene pool, but not always. Don't go by that rule. Instead, use this rule. Dominant gene means that what shows up in a heterozygous organism. So, for instance, above, if you were HbA, HbS, we would ask, do you have normal hemoglobin or do you show signs of sickle cell anemia? That would tell us what the dominant allele would be. So in this example above, the HbA is the dominant because individuals are what we call carriers. They can carry this combination, they carry the sickle cell anemia trait, but they don't show any signs of the disease. They make enough normal hemoglobin that their cells don't sickle and turn into things that block blood vessels and cause the disease. Alright, that's my intro for this week. Next week I'll be getting into the Hardy-Weinberg equilibrium. And if you have any questions, email me, contact me somehow. Thanks a lot.